Good morning. I am Eirini Velliou. I'm professor of bioengineering and I'm a principal investigator at the Center for 3D Models of Health and Disease in the Division of Surgery and Interventional Sciences at UCL. So within our center, we are particularly focused on using bioengineering approaches, biomaterials, bioreactors and their combination, to design in vitro tissue mimicries. We work with a variety of different tissues, and many of us work in cancer as well. So we're trying to create advanced models of different types of cancers for in vitro studies and in vitro drug testing. Okay, I'm a bioengineer, and within my research group, we are using bioreactors, biomaterials and their combination to study how different biological systems grow in the lab, grow in vitro, how they interact with each other, as well as with our environment. Within that frame, we are particularly focused on tissue engineering, wherein we fabricate scaffolds with different biomaterials, both synthetic and non-synthetic, and we load those scaffolds, if you like, with different cell types of the tissue environment. We also use bioreactors, like the Kirkstall bioreactor, where we introduce dynamic flow, which can mimic the interstitial flow that occurs in tissues. And within this context, we are trying to understand how different factors of the tissue microenvironment affect the way cancer evolves in vitro, as well as the way cancer could respond to novel and conventional therapies. So for a number of years, we've been focused on pancreatic cancer. Pancreatic cancer is an aggressive cancer with very low survival rate and a very complicated tissue or tumor microenvironment. So what we do is we try to recreate in the lab complex multicellular tumors of the pancreatic tissue, when we have cancer cells as well as other cells of the tumor, such as activated stellate cells, fibroblasts, or endothelial cells, that we know interact with the cancer and they help the cancer progress. So we've developed such a spatially advanced multicellular model of pancreatic cancer that can mimic the fibrosis, or else known as desmoplasia, that occurs in the pancreatic tumor. And we are using the Kirkstall device to see how dynamic flow and shear stress could affect the way the cancer cells, but also the non-cancerous cells, so the stromal cells of our pancreatic cancer model, evolve, progress in vitro, and also whether introduction of flow could affect the way they would respond to chemotherapy. So the survival rate is very, very low, and it's less than 10%, and unfortunately it has barely improved really since the 70s. In contrast to many other cancers where we've seen massive improvements both in detection as well as in treatment approaches. So there's definitely urgent need for much more research into pancreatic cancer and into the discovery of novel treatments for pancreatic cancer. But for that to be done, we do need, next to animals, definitely animal-free approaches and high throughput systems that could help us screen the progression of the disease and its response to treatment. And tissue engineering is one route on that direction. Pancreatic cancer at the moment is the fourth leading cause of cancer-related deaths worldwide. So using our multicellular compartmentalized scaffold, where we can mimic different levels of fibrosis or desmoplasia, which is a hallmark of the pancreatic cancer disease, we immerse them into the Kirkstall bioreactor in the individual chambers of the reactor, and we introduce dynamic flow. So what we do is we introduce flow for different time periods, and we observe changes in the cellular behavior, in the matrix deposition, in upregulation of markers that are linked to aggressiveness of disease as a function of the flow and the presence of the flow. Interstitial flow is an important feature of the tissue microenvironment, and it could be present at different levels in terms of the interstitial flow rate, and therefore investigating that could help us better understand how that feature, which occurs within the body, which is present within the body, could potentially affect the evolution of the cells in vitro. The results we've generated so far with the Kirkstall bioreactor through a project that was funded from the Medical Research Council show that the dynamic flow does affect significantly the evolution of the cells. It does affect the aggressiveness of the cells. It does affect the response to treatment, as well as their progression in vitro. Our Medical Research Council project is soon coming to an end, and we are hopeful that we will be publishing our results on these exciting findings on using pancreatic cancer models under flow within the next few months.